Welcome back in lecture series on topic electrochemistry. In last video, we have studied molar conductivity and relation between conductivity and molar conductivity of strong as well as weak electrolyte solutions. And today in this lecture, we will study how conductivity and molar conductivity is measured experimentally using Wheatstone bridge method. You know that conductance of solution is a reciprocal of resistance, and therefore the conductivity of solution is determined from the resistance of solution by Wheatstone bridge method. We also know that unknown resistance of any metallic wire is measured on a Wheatstone bridge by directly connecting it in Wheatstone bridge. But like metallic wire, we cannot connect electrolytic solution, and therefore we cannot measure the resistance of the solution like metallic wire on Wheatstone bridge, and therefore for that purpose conductivity cell is used. So first we will study in this lecture what is meant by conductivity cell. Then cell constant means what? Then determination of this cell constant, and then determination of conductivity, and from conductivity how to determine molar conductivity. All these points we will discuss in this lecture. Determination of molar conductivity consists of three steps. First step is determination of cell constant. Second step is determination of conductivity of the given solution. And third step is calculation of molar conductivity. So the conductivity of solution can be determined from resistance measurement by Wheatstone bridge method. So what is meant by conductivity cell? That we will see first. Conductivity cell is like this. It consists of a glass tube. This is the glass tube like this. And this glass tube is fitted with two platinum plates like this. And these two platinum plates are separated by distance L. And these two platinum plates are coated with a thin layer of finely divided platinum black. And this layer of platinum black is obtained by electrolysis of chloroplatinic acid. Solution of chloroplatinic acid. Important point, note down this because in short answer question or in MCQ, question can be asked on this point, that is, name the solution by which the finely divided platinum black layer, thin layer of finely divided platinum black is obtained. So it is obtained from chloroplatinic acid. And then this cell, whole assembly of the cell, this is called as a conductivity cell, is dipped in a solution whose resistance you have to measure. So this is called as conductivity cell. Conductivity cell is it consists of a glass tube with two platinum plates, and these platinum plates are coated with a platinum black, finely divided platinum black, which is obtained from electrolysis of chloroplatinic acid, and the whole cell is known as a conductivity cell, and it is dipped in a solution whose resistance you have to measure. So conductivity of the solution is equal to the relation between resistance and conductivity we have studied. So K, that is kappa, is equal to 1 upon R into L upon A. L means the distance between these two platinum plates, that is how much? L. And A means the area of cross section of these two platinum plates. So K is equal to 1 upon R into L upon A. And for given conductivity cell, the ratio of separation between two electroplates or two electrodes, that is L, divided by the area of cross section, that is A, of the electrode is called the cell constant. So L upon A is nothing but cell constant. So here length is measured in SI unit in meter, and the area of cross section is measured in meter square, and therefore unit of cell constant is meter inverse. SI unit of cell constant is meter inverse, and in common unit is centimeter inverse. So conductivity is equal to 1 upon R into L by A. But this L by A is nothing but cell constant. So once you know the cell constant, you can calculate conductivity by this relation. K is equal to cell constant divided by resistance. That is nothing but what? Conductivity of the solution. So see here how to determine the cell constant. So cell constant of given conductivity cell is obtained by measuring the resistance or conductance of the standard solution. And the standard solution used is 1 molar or 0.1 molar or 0.01 molar KCl solution, because its conductivity is accurately known at different temperature, and therefore it is taken as a standard solution. So resistance of this KCl solution is measured by Wheatstone bridge method. For the measurement of unknown resistance, you know that Wheatstone bridge method is used. And a KCl solution of known conductivity, whose conductivity is known to you, is taken in a beaker like this, and conductivity cell is dipped in it. And Wheatstone bridge consists of a wire AB of a uniform diameter, and in one arm of the Wheatstone bridge, variable known resistance, that is Rx, is placed, and the two electrodes of this conductivity cell are connected to the other arm of the Wheatstone bridge. And here the current detector is connected to these two arms. And to this current detector, this wire is there, that is called as a sliding contact, and it moves along the wire AB. And both the terminals of this wire AB are connected to the alternating source. This AC represents the source of alternating current. There may be a question in exam, that is why DC current source is not used. So why DC source is not used? Because it causes electrolysis of electrolyte, that means composition of electrolyte may change, and due to which concentration of the solution changes, and therefore DC source is not, direct current source is not used. Instead of that, here alternating current source is used here. When current flows through this circuit, then the here this current detector shows the deflection. Then this sliding contact, that is F, is moved along the wire AB from A to B until no current flows. That means here no deflection is observed. That point is called as a null point or balance point. So this sliding contact is moved on this along this AB wire and null point is obtained. Suppose it is at point C. Then the length AC is measured, and that is proportional to the resistance of the solution inside this beaker. And this BC represent what? The resistance of this Rx. So resistance of conductor is directly proportional to its length according to Wheatstone-Bridge principle. And therefore resistance of solution is directly proportional to length AC, and resistance Rx is directly proportional to length BC. This one. And by measuring the length AC and length BC, and by knowing the value of Rx, that is variable known resistance, the resistance of KCl solution can be calculated by using the formula resistance of solution divided by length AC is equal to Rx divided by length BC. That means here resistance of solution is equal to length AC divided by length BC into Rx. So cell constant is equal to conductivity into resistance of solution. So from that cell constant is obtained. Now how to determine the conductivity of the solution, that we will see. Now here the KCl, once you calculate the cell constant, then KCl solution is replaced by the given solution whose conductivity is to be measured, and its resistance is measured in the same way as that of the KCl solution. And from that you know that cell constant is equal to conductivity into resistance of solution. So conductivity of the solution is equal to cell constant divided by resistance of the solution. So using this formula, conductivity of the given solution is calculated. And the molar conductivity of given solution is determined from this formula, that is lambda, that is molar conductivity of given solution is equal to 1000 into conductivity divided by C. So using this formula we can calculate the molar conductivity of the given solution. So thank you.